Hello biology students, and welcome to our new unit on DNA structure and replication. So in this video, we're going to be just looking at one thing, which is this first question here, which is how do we know DNA is the hereditary material? So in other words, what experiments were performed, and how did scientists ultimately come to the conclusion that DNA carries our genetic information and not something else? Okay. So a little history about what was known before scientists discovered that DNA was the heritable material. So it was known for quite a while, actually since the late 1800s, that genetic information or heritable information was carried in units called genes, okay? And also, at this point, scientists knew that genes made up these structures called chromosomes, okay? However, the big problem was that scientists also knew that chromosomes had two pieces to them. Their chromosomes are made up of DNA, deoxyribonucleic acid, but they're also made of protein. Now remember we talked about earlier in the year that in a chromosome, a eukaryotic chromosome, DNA is wrapped around the histones, those proteins that kind of help to hold the chromosome in its shape. So this was a problem for scientists because they knew that the genetic information was carried in chromosomes, but they didn't know if it was the DNA piece that was carrying the genetic information or if it was the protein piece. So until about the mid-1940s, so really not that long ago, scientists pretty firmly believed that protein actually held genetic information, and DNA was basically useless. I mean, they really thought that protein held all of our genes. And there were a lot of reasons they thought this. First of all, proteins are very, very varied. So remember, proteins have 20 amino acids. They can make millions of combinations. We have tons of proteins. Proteins have specific jobs. They can do all sorts of stuff. And really, scientists didn't know much about nucleic acids. They thought they were pretty much not useless, but there's this big, strong, awesome molecule, the protein, that can do all this stuff. And then there was the nucleic acid, which seemed very lame in comparison. So it wasn't until, like I said, about the 1940s, 1950s that, through a series of experiments, that scientists realized that DNA was the heritable material, not protein. And there are a whole bunch of scientists that we're going to talk about today that are listed here that did experiments to help figure this out. Okay, so the first guy we're going to talk about is Griffith, Frederick Griffith. And he did experiments using mice. So I really like this picture here because it does an excellent job summarizing his experiment. So what he did, he worked with mice and bacteria. Actually, the bacteria was called Streptococcus pneumoniae. It's the bacteria that causes pneumonia. And he had two different versions of this bacteria. He had what was called the R strain, and he had what was called the S strain. These are just different versions of the pneumonia bacteria. So what he did, he took the R strain version of the bacteria and he injected it into the mouse, and the mouse ended up living. So he concluded that the R strain of the bacteria does not cause pneumonia. Then he took the other version of the bacteria, he took the S strain, he injected that into his mouse, and the mouse died. So the mouse got pneumonia and dies. So his conclusion here was that the S strain does cause pneumonia. So this, nothing about this is revolutionary. But then he did something kind of funny. He took the S strain bacteria, so this is the deadly bacteria, he heat killed it. And that means he took the bacteria and he heated it up and basically killed the bacteria. He injected that dead bacteria into the mouse, and the mouse lived. Okay, so if you have deadly bacteria, that does not cause pneumonia. But then, the fourth part, and this was the real big shocking part of his experiment, he combined the harmless form of the bacteria, so he took the living R strain, which was harmless, and he took dead S strain, which remember is also harmless, and he injected these two harmless things into the bacteria, and then the mouse died. So this was very confusing for Griffith, and this was actually a very revolutionary and interesting conclusion. So let's look at the next slide about this a little bit more. Okay, so to summarize a little bit, so Griffith was working with two strains of Streptococcus pneumoniae, Remember, the S strain he determined was pathogenic, aka it's a pathogen, it can cause pneumonia, it's deadly. And then he also discovered that the R strain was harmless and did not cause pneumonia. However, when he took dead S strain, which should be harmless, and live R strain, which would also be harmless, when he injected them into the mouse, this caused pneumonia, this caused death, and this was very, very bizarre. Okay, so Griffith's big conclusion, and after he thought about this for a long time, he basically concluded that there must be something in that disease-causing strain, and even after it was dead, that disease-causing agent was able to be transferred to the living R strain, okay? So something in the dead harmful bacteria changed the harmless living bacteria, okay? In other words, we say that the living R strain was transformed by something, by the genetic material that was released by the S strain, okay? And something kind of cool, all descendants, so all the offspring of the R strain bacteria could also cause the disease. So even though the R strain is supposed to be harmless, it was changed, it was transformed. So now it can cause disease and all its offspring can cause disease. So we call this a transformation, when you get a change in your genotype and phenotype due to the fact that you incorporate outside DNA. So now we know that the harmless bacteria picked up DNA from the dead harmful bacteria, but at the time Griffith didn't know this. He just knew something was transforming it. And scientists kind of looked at his conclusions and said, well, it might be protein and it might be DNA that's being transferred, and we're really not sure. Okay, so the next set of experiments happened almost 15 years later, so quite a bit of time later. And this was 3 scientists. So Oswald Avery was kind of the lead scientist, and then he had 2 colleagues, McLeod and McCarty. Okay, and they wanted to do a follow-up on Griffith's experiments. They wanted to figure out what in that dead harmful bacteria was transforming the harmless R strain. Okay, so basically what they did, they took the S strain that Griffith used, and they wanted to determine which macromolecule was transforming or changing the R strain. Was it carbohydrates? Was it lipids? Was it proteins? Was it nucleic acids? Okay. So they did this they basically isolated one factor at a time. So they isolated the carbohydrates, isolated the lipids, isolated the protein, or isolated the DNA, and they tried to figure out which one would actually transform the bacteria. So let's look at the steps of their experiment. Okay, so basically here, Oswald and his colleagues have a tube that's full of the heat-killed S cells. So this is the killed harmful bacteria. And what they did, they separated out, they took the lipids out, they took the carbohydrates out, and then they also separated the protein, RNA, and DNA. Okay, so they have all these things separated that came from the dead harmful bacteria. Okay, so then what they did is they separated them all out. So now they have a one sample that just has the DNA and the RNA from that heat-killed S. They have one sample that has just DNA and protein, and they have one sample that has just RNA and protein. Okay, so in each one, they eliminated one thing. So they eliminated the protein, eliminated the RNA, and eliminated the DNA. And then they added this stuff from the heat-killed S to the R strain. They added it to the harmless strain, the strain that won't cause pneumonia, and they looked to see which one was transformed. So when they didn't have any protein, when they just had DNA and RNA, the cells got transformed and the mouse died when they injected it into a mouse. When they didn't have RNA but they still had DNA and protein, a transformation happened. They injected it into the mouse and the mouse died. But when they were missing DNA and they added that to the R strain was not transformed, and when they injected this into the mouse lived. So in other words, it's only when DNA is present that a transformation will happen, okay? So this was huge. They were so excited about this. They were like, yes, now the whole scientific community is going to believe that DNA is the genetic material, and even though everyone currently believes it's protein, we've proved them wrong. However, when they, published their paper, basically no one believed them. Everyone believed that, their experiment must have been faulty. There's no way that DNA could possibly be the genetic material because it's so lame and protein is so much cooler. Okay? Okay, so then a few years later, in fact, almost 8 years later, so it's been almost a decade, and people still don't believe this experiment. We had one last set of scientists who did a final experiment showing that DNA is their heritable material. So these two scientists, Alfred Hershey and Martha Chase, they used a different organism, or a different vector to study this system. Instead, they used a virus. And a virus isn't actually an organism. A virus is not living. Okay? So viruses, they share some characteristics of life with living things, but they're not actually alive. So they're not actually cells. And the reason they're not actually cells is because they're only made up of two things. They're made up of a protein and a nucleic acid. So they're very simple. They're not complex enough to be cells. They're also not cells because they cannot reproduce on their own. Okay, they have to hijack other cells in order to reproduce. This is what a virus looks like. This is a cartoony picture right here, and this is an actual microscope picture. So they're very weird looking. They kind of look like aliens. And you can kind of see they have this outer coat that's made of protein. They have a little shaft, and they have little like leg things. And this whole thing is made of protein. And then inside this capsule up here, they have their DNA. So the black part here is all protein, and that red part is their DNA. And the way they reproduce is they land on the surface of a cell. So here's a cell I'm going to draw. And they literally kind of like use this part kind of like a little needle, and they inject their DNA into the cell, make more copies of their DNA, and make more copies of their protein coat. And basically, they reproduce so much inside the cell that it fills the whole cell and the cell bursts open. Okay. So this T2, the bacteria, it can reprogram the cell by injecting its own DNA into the cell and then forcing the cell to make more copies of the virus. Okay? So now Hershey and Chase kind of figured, okay, if we can figure out if the virus is injecting its protein or its DNA, we'll know which piece of the virus contains the genetic material. Okay? If it was injecting its DNA, that means the DNA must have the genetic material because that's the part that hijacks the cell. And if it injects its protein coat, then the protein coat must be the genetic material because that's how it's reprogramming the host cell. Okay? So in order to trace where the protein and DNA was going, Hershey and Chase used radioactive isotopes. So basically, remember that proteins have sulfur and DNA does not. So basically, the sulfur that they used to make the virus protein coat basically glowed yellow. It was radioactive. So that way they can look at the protein, they can look at the virus, and they can watch where the coat went. Wherever the glowing yellow color went, that's where the protein went. And remember that DNA has phosphorus and protein does not. So they used radioactive phosphorus to track where the DNA went. You know, wherever the green color went, that's where the DNA was going. So they can figure out what was going where. Okay, so here's their experiment. Let's actually look at it. So here they have basically a vial that has a bunch of cells that the virus are going to infect, and then a bunch of virus. And these viruses have been grown with radioactive sulfur. So basically their proteins are going to be glowing. So you can see here the protein part of the virus is glowing yellow. Okay? So they allowed the virus to infect the cell. And then they actually put them in a blender. And what the blender did is the blender mixed up and agitated the cells in the virus. So basically the coat part, like whatever was not inside the cell, got separated from whatever was inside the cell. They then put it in a centrifuge, which is a machine that basically spins really fast and separates things by density. So things that are really dense, like the cells, end up at the bottom of the tube. And things that are not dense, like the liquid, end up at the top of the tube. So you can see what happened was that the top of their tube was glowing yellow, which suggests that the protein is not inside the cells, because the denser part, the cells, were not glowing yellow. So if the protein did not go inside the cell, that means the protein's probably not the genetic material. Then they repeated their experiment, but this time they had virus that was not labeled for protein. Now the virus is labeled for DNA. So now the DNA is radioactive. So they do the same thing. They let the viruses infect the cells. They put it in a blender to separate the virus from the cell. And then they put it in a centrifuge, and this time they saw that their cells were glowing green, which implied the DNA that was green was inside the cell. And if the DNA was inside the cell, that means the DNA must be the genetic material of the virus. So then, finally, this experiment really concretely proved that DNA was the genetic material and not protein. So finally, the scientific community started to come around after this was published and realized that DNA was the heritable material. So now the only problem with this, at this point, no one knew much about DNA at all because no one had studied it because no one thought it was important. So after this, there was a big race to figure out more about DNA, which we'll talk about more in our next video.